in the context of today's meeting, we in the UK have always tried to include as many people from different walks of life as possible. But now we need to go further. We need to think more about international aspects and how we can reach across borders that unfortunately recently have gotten even more difficult to breach. But we will not stint until we have such close connections across different countries, because in genetics anyway, what we need to do is to ensure that our data have the power to find the results, the discoveries that matter to people. And for that, we need the power of data, and for the data to be shared. So without any more from me, I'm just going to introduce Marte to you, and looking very much forward to your talk today. So can I use the arrows?
Thank you. Thank you so much for inviting me. It's a real privilege to come here and meet you all, both online and in person. Yes, so I'm going to talk a little bit about the immunogenetic studies we've been doing for the last couple of years. So just a little bit about us first. So I'm from the research group in Professor Benedicte in Norway, doing genetics in autoimmunity. And I am a project group leader named HLA disease, but I'm the one doing the ME research mostly. We've had 2 PhD candidates and 2 Master of Science students. They have all just finished. And then we have Anne Ridland, that's actually working on a different disease, but she also have an ME project because she was hired on that project previously. So we're situated in Oslo, and I work at 2 places. The Department of Medical Genetics, where my main time is spent, and then also at the Department of Immunology at Viken. So I have both a foot in immunology and genetics, which is great for this type of research. And please stop me if I'm talking too fast, because I have a tendency to do that, and just interrupt me if there's something unclear. So I wanted to talk a little bit about how we started doing ME research, because like I said, me and Benedicte, we come from the field of autoimmune diseases, complex diseases. And we were astonished by the debate climate in Norway. And from the excellent talk just now, it's been obvious that it's the same in the UK. It's been really harsh, the debate climate, especially in the media, with a lot of not well, I mean, when clinicians stamp patients as activists, you don't get a good collaboration climate for research. That's not being inclusive. So we were very astonished, and we wanted to move into the field of ME. And we had a great research group in Bergen doing rituximab ME. They were doing intervention with drugs. So we contacted them trying to see if we could do some genetics work. And they helped us get some private financing from the Causal Trust, which makes cheese on tubes in Norway. And they financed a PhD project for us for 3 years. So that was limited funding, so we had to limit our research. So we started with the HLA genes, and I'll explain what that is later for those not knowing them.
I was just wondering why you got interested in ME in the first place.
Because it's obvious to us that it's a complex disease, and we looked into research and was so little done on genetics, and we couldn't understand why people didn't embark on doing research on this disease, because it's affecting so many. So that was actually the basis for it. And then we got the funding, and that gave us some temporary results. And then the government in Norway, the Research Council of Norway, decided to have a specific call for any research later on. So we had already started, so that was excellent for us. And it was called Behov ME. And Behov, it's needs. So what it reflects what type of needs do the ME community think we should research on. So that was the first time they tried this public involvement in Norway from the Research Council, where they told everybody whoever in the public could submit what type of research they thought was needed. Then they had a committee looking at it, and people applied, and they selected some of the research projects to move forward to the normal kind of evaluations. And we were lucky to be one of 4 that got funding for that. And we, of course, then had, when we formed our application, we had patients with ME giving us input on what type of research we should do. So our working hypothesis have always been, is ME, CFS, an immune-mediated disease? That was the big question for us. So our aim was to investigate immunological genes, and then also the potential involvement of the immune system. So we went for the more invasive way of taking samples from ME patients, which is blood. We hired a PhD student with a medical degree, and he was trained by another doctor to also visit some of the patients. So most of the samples were taken by whoever healthcare professionals they had, the medical doctors, but we also had a specific focus on getting more of the severely ill. So our PhD student actually went around visiting some of them. And then, of course, we needed DNA, and for some of the samples, we have serum or plasma or even RNA as well. So that's the benefit of using blood for us. So I'm just starting with this. We have, like I said, it's nowhere near the decode ME, and that's also why I feel it's a privilege to be here. I love the plans for decode ME. I mean, I'm a geneticist, of course, numbers matters. But this was the largest at the time. It's all Norwegian samples. There's a little bit of different numbers that has to do with some samples falling out, some coming in, not having enough DNA. But I mean, we had more than 400 Norwegian samples, which is very small compared to, but I mean, when you look at the previous genetic studies, or most studies in ME at this time, was around 40 to 50 people involved. So I said I'd come back to the HLA, which is a very important gene complex. So the human major histocompatibility complex, and the name comes from the field of transplantation. So I mean, that's has driven the field and research of these genes. They are really important in immunological, and the HLA stands for human leukocyte antigen. They are highly polymorphic, and with that, I mean they vary. There's like thousands of variants of each gene, and that's important for. I mean, these have a normal function for us to be healthy, to combat different viruses and bacteria in normal life. But they are medically important also in transplantation, and they are associated with autoimmune diseases. So there's hundreds of diseases associated with these genes. We typically divide them up in class 1, 2, and 3, and what we've been researching is the class 1 and class 2. And I just put this up here to show you a bit about the HLA variation, because when we talk about HLA variation, it's that different genes. So you can see the HLA-B gene has more than 8,000. I think it's actually up into the 9,000 variants at the present time. These are cell surface molecule, and they have what we call a peptide binding group here, up and to the top. And the class 2 genes are encoded by 2 genes in the MSC. So you have the DQB beta will encode one part, and the DQ alpha 1 will encode the other part. And those together will make up the peptide binding group that presents to T cells. So, like I said, we looked into what had been done, and I must say, I've been very generous here with what you call ME. I mean, none of these diseases are actually noted as ME. It's the chronic fatigue syndromes. I haven't, I mean, what type of classification they've done. I mean, it's very broad, and it goes back to the 90s. And the large one was 110. They didn't do all genes. It's the methods used are very broad, so you don't get the detail of what type of molecule you're actually looking at. Could be serological instead of using genetic methods. So there were some of them showing some signs, but I mean the numbers were very low. So based on this, we knew that we weren't expecting like a really, really strong sign, because in other diseases where you have that, you would see it with these numbers. But there were some signs that gave us promise that with more than 400, we might be able to see something. So we went for high resolution sequencing to do the genotyping. And this is actually the same that we do for bone marrow transplantation matching. So it is actually the best type of typing you can do at the time. So we got really detailed knowledge about which variants of these genes, and I'm not going to go into the details here about it. At the first time, like I said, we have limited funding, so we were lucky to have control data set for 6 genes. We typed the ME patient for all the 11 classical HLA genes, but we then, since the controls only had for 6, we could only do those analyses in the beginning. Our patients were the mean age around 40 years, predominantly women, as you all know. More than 60 had the disease for over 10 years or less. And we had 41% that was bedridden or strictly housebound. And that was increased by our PhD student, Asgeir Lande, who's the first author on this paper, because the Norwegian samples were collected in 3 parts. It was from the Rituximab ME study in Bergen. Then we had the CFS ME Center that do a biobanking. And then we had more than 100 that we collected ourselves. And he, like I said, he actually had a centrifuge in the back of his car, and he went around the Oslo area to the very ill patients and like wearing no perfume, taking really care to minimize the stress, and got blood samples from more. So we had an increase in the percentage of the severely ill in our data set. A little bit more about the patient characteristics. Like you see, I mean, for me, this kind of indicates why ME might be what we call an immune-mediated disease. It's a female predominant disease. There's a lot of first-degree relatives with autoimmune disease, which you see in these type of complex diseases, that you have relatives not maybe with the same disease, but with an autoimmune component. So when we did the study, we found 4 alleles showing a positive association with ME. And it was specific variants of the HSC, B, and GQB1 gene. We also had some signs of negative association. The significance level isn't like genome-wide association significance. I mean, we couldn't expect that at the numbers we have. But in this study, they survived correction for the number of tests we did at the present time. So this might be a little bit messy slide, but it's to show that what we could see, that it looked like 2 positive association signals, because, so I'm not sure if people online can see this, But these two make up what we call a haplotype, so they are inherited more often than what you would expect together. So we can't say which one is the primary associated of these. The same for these one. So what we ended up concluding is that the C allele and the DQB allele tag the HLA associations, but we couldn't distinguish if it was one or two signal. And if it's one signal, it could be something else than these genes, because this region contains hundreds of immunological genes that are not called HLA, but also are important. So we then had a new PhD student arriving, and we added SNP, single nucleotide variants, throughout the genome, but with an immunological focus. And then we used more than 5,000 of these that were covering the HLA region and analyzed them together with the HLA. And here I must just make you aware that we now are using different controls. Because by that time we had a collaboration to get with somebody else to get our 500 or around 500 controls HLA types, which were also had SNPs, like so we could combine. And that gave us the possibility to try to fine map. So with fine mapping, that's trying to pinpoint where is the signal coming from. So in color are the HLA, either as allelic or analyzed as amino acids or per locus. None of these association signals reached what we call the genome-wide association signal, which all geneticists would like. But like I said, we didn't expect to either, because from the HLA first study, we knew kind of what significance level we were at. But what this made us look at, so we could then try to do conditioning analysis. So looking at if we condition on the HLA-C, do the signals in the DQB1 region disappear? And they didn't. And the opposite, if we condition on the DQB1 on the right here, do the signal in HLA-C region disappear? And it didn't. So that told us that it was most likely at least two independent signals. This analysis also shows that around the DQB1, it seems to be the DQB1 that is the main culprit, maybe. But around HLA-C, we're quite unsure if it's HLA-C or another gene in that vicinity. So I'm hoping that Decode ME could later give some clear picture into this. So, like I said, I mean, the goal is to try to find causal variants. This region is very difficult. And I mean, still in the other diseases like type 1 diabetes and narcolepsy, and these, we still don't have the full picture. And I mean, we're lagging years behind. But we do think that DQB1 might be the main gene in this signal association. But we are not sure if it's actually the DQB1-0303 allele, or if it's the amino acid with the aspartic acid. Because what you see here is that most alleles encoding that amino acid, the most common ones are increased in patients. And if you have the alanine instead, most of them are in general decreased in patients. So, and this amino acid is kind of a key amino acid in the molecule in the peptide binding group, and it seems to be have some involvement in the stability of the molecule. So it's will be interesting to look further into. So I've been talking about HLA and autoimmunity, and I don't want to put ME into the perspective of other diseases, because a lot of autoimmune diseases have strong HLA associations, but not all. And like rheumatoid arthritis, it's not that all rheumatoid arthritis patients have an HLA association. It's the antibody positive one that's driving the HLA signal. And I put down ME at the bottom with the DQB1. So DQ9, that's a serological way of saying DQB103. So the odds ratio, so the risk here, is not as bad. I mean, people say it's low, but I mean, myasthenia gravis is it's a rare disease, but it's a well-established autoimmune disease, and that also have the same, and nobody questions that. But you can see it's a bit different when you split into subgroups, which could be something within ME as well. I mean, we couldn't split them into subgroups. That's something that's very possible when you get larger numbers. But I must say that most carriers of HLA risk are actually healthy. So when we, as researchers, present this graph, where you see that the carrier frequency is increased in patients, we must remember at the bottom here, the number of individuals are different. And that's important. It's easy to forget when you see us researchers present. So that leads me to, are these associations useful for diagnosing? And if you look at if you think the blue ones are people carrying the risk variants, most of them will never get disease. It's maybe just 1 or 2 unlucky people. So, like I said, we worked together with the research group in Bergen doing treatments that they did rituximab and also cyclophosphamide. And I just must say, yeah, it's a really harmful drug that has a lot of bad side effects. Nothing that's used by these doctors very loosely. But what was surprising that what we saw, and also here on right, I mean, you can see that both there's a high numbers of responders, but within the risk positive, 83% was actually carrying the risk alleles. But this, I would just must stress, is a very small open label study, so it's prone to the placebo effect as well. And it will need to replicate our HLA findings, and it should be performed a double-blind study if ever. There are concerns with this drug because it has a lot of side effects. I mean, females can actually become infertile. So I will not recommend to do HLA testing and start this type of treatment, just so that's stated. So why are we then doing this? I mean, we do our research for patients, so if you can't use it diagnostically, why do it at all? But with genetic studies, also ours, we can pinpoint to biological roles where to look further for potential treatment. So with the HLA, you can either go to the cytotoxic T cell or the T helper cells, and then in our case, we had associations with both, so we might have to look at both roads. Also, there's some genes encoding receptor called KIL, so it stands for killer immunoglobulin-like receptors. And the HLA-C is actually a ligand for those as well. And NK cells, natural killer cells, I know people with them in research, and I know has been part of the picture for the research for a long time. So then what about the T cell receptor? Because that's on the other side of the HLA. The HLA presents to the T cell receptor. So we did a small study, also due to limited funds and resources, because Schlauch et al. Had a publication where it showed a great significant association with the T cell receptor alpha locus. Their allele frequencies were very deviant between cases and controls. Ours, and we also looked in the UK Biobank, were not, so there were no association signal for ours. And if you look on all the controls, also the Schlauch, they are all at the similar as the 1000 genome. So what's very deviating from the rest is the cases in the Schlauch et al. Paper, and they had 42 cases only, which are prone then to selection bias or something like that. Then I'm trying to finish up. Genetic associate, we also did the rest of the genome with an immunological focus. And we didn't find any signs of genome-wide association signal, studies signal, but which is the red line. But we had signals in the suggested region. And this is the TPPP gene, which had several SNPs showing that it could be associated. The main one has low frequency. It's a low frequency allele, so that also makes it prone to selection bias. But this would be interesting to see in a larger study later on. Yeah, and this is just to show you that TPP, I mean, the Norwegian one was varying that signal the most, and the Danish one was deviating from this. So in summary, our studies indicate a potential role for HLA genes. Our significance level is limited by the numbers of individuals we have. But we think that our results indicate that the immune system is involved in ME, and we're really looking forward to further studies. And our ongoing and planned projects are, we have here, so the killer immunoglobulin receptor gene project going that needs to be finished. We also have a project on extracellular vesicles, which are cell-to-cell communication with signals, where we have some interesting preliminary data at the moment. And we just got funding for this year for a new PhD student that we will hire before Christmas, continue our research. So then I would really like to thank all the patients especially, but also the controls, because controls are all equally important to do studies in this. So, and of course, all the fundings and all of my collaborators. Thank you.
Do we have questions in the room and online? Joshua.
Sorry to introduce myself. Sorry to introduce myself online.
Please do.
Okay, so Joshua Dibble, University of Edinburgh. So thank you, good talk. Can I ask about HLA genes?
Yeah.
You said the coincidence with autoimmune conditions was about 15%.
What did you say?
So the coincidence of NCFS and autoimmune conditions was about 15%. Did you check that the people with your HLA genes don't have other autoimmune conditions?
So what we checked is that those other autoimmune diseases are not associated with those alleles.
Okay, yeah,
Yes, so that's what we did, because that's quite important. Yeah, so that's a good question. Thank you.
Some questions online.
We do, yes. The first question was, how did you recruit patients with severe ME who don't always have access to the internet?
So we got help from the Norwegian ME Association, Patient Association, to advertise. So that was typically through their caretakers. So would be parents or relatives. So they could contact us. And we, at that time, it was, our questionnaire was actually in paper. So the caretakers were allowed to help fill out on on their behalf. So that was the way we did it. And also we had, the ME Association had an overview of some of the very severe, so they kind of knew that, so they might have made them especially aware. So I think it might have been different ways, but a lot of thanks to the ME Patient Association to get to those. That was excellent.
What was the selection criteria that you used?
Oh, I forgot to say, the Canadian criteria. Yeah, consensus criteria, yes. And we're quite strict on using those, which has been a great debate in Norway. We have some strong voices in Norway, especially some of the sides of the debate, which thinks that you could use any criteria. But my opinion on that is when it comes to care, I think like clinical care, the criteria used doesn't matter because patients, regardless, should get good care. But when it comes to genetic studies, it might be a bit different. So, I mean, it's not one answer for all, but for us, we use the Canadian criteria. But of course, the UK Biobank is self-reported for data from that.
Self-reported diagnosis.
Yeah.
Second question was, can you explain the genome-wide association study?
So the genome-wide association study. So it's not a pure genome-wide association study because it's restricted to immunological genes. So it has a bias for genes involved in autoimmune disease, is the one we used. But it's simply put, you do genotyping throughout the genome with single, so genetic variants that just one base different, and you look at the association difference or the allele frequency difference between patients and controls. So it's not at the individual level, it's at the population level. And so we can't use it diagnostically, but it can give an impact on which pathways are involved, which is the main goal for our study at the moment, but not later down the road. So.
What was the selection criteria for the control population? Because I know ME/CFS has that male-female bias. Did the controls have that?
No, we don't have that, and it's actually quite healthy people because they are blood donors, and actually not blood donors, they're also bone marrow donors. So there are a lot of so it's actually a selection for a very healthy population, but we shouldn't do studies on altruism on those because they are really people that give back to community. So yeah.
Graham Kevin, University of Edinburgh. I'm wondering what your HLA results mean in terms of the mechanistics of the disease. The HLA obviously affects which peptides you can present from your HLA to the T cells. I'm wondering whether you think it means whether there are implications on susceptibility to viruses that could be an initial trigger of the disease, or whether you're thinking that it might predispose to an autoimmune reaction in response to that, and whether there's anything known about how these particular alleles affect the susceptibility to viruses that have been potentially found.
First of all, very good question. And the quick answer is, I don't know. I have no idea. I think it could be both. I've worked with a lot of different diseases. We still have no clear idea about celiac disease. We know the gluten peptide there, but we still don't know what's the triggering environmental factor besides gluten. So what's making people sensitive to gluten? So I think it's impossible for me to say at this present time. I think it could be both what you present, but it could also be how susceptible you are to certain infections. They can also be, if you have some alleles, you might not be as easily infected by it. So, yeah, that's the interesting part and difficult part. Yeah.
In your signal, did you see any difference between male patients and female patients?
No, we didn't.
Joshua.
Can I ask about the model you used? Was it linear logistic? What kind of variants?
Yes, it was plain linear logistic.
Okay, for the HLA alleles as well.
Oh, you asked this previously. No, I didn't run the analysis. So I don't remember, but we use on the HLA we use an EM algorithm on the first paper at least. Yeah. So yeah, it's actually a program from a guy in the UK. So yeah.
Do we have any further questions online? One quick one.
Yes, what drug was trialed, please? Let's catch it.
That wasn't us, that's our collaborators in Bergen. So they used rituximab. So that was their large study. So they did 2 drug trials, one with rituximab, and then they did one, and these are published, and they don't see anything. And then they did a large double-blinded on the rituximab. They also had this small open label with cyclophosphamide. So that's cyclophosphamide, and both these are cancer drugs. But the rituximab is also used in rheumatoid arthritis in lower dosage. So it has to do with the immune system and the cells there. But these are, like the doctors there say, you shouldn't do this without, like, out of clinical trials because they have severe side effects. So they had really close, follow up on all these patients. These are published. It's Fluge and Mella in Bergen. Dr. Fluge and Mella.